Good evening, everyone. I'd like to welcome everyone to the Greenwood Genetic Center's Science on Tap educational program. We are very happy to begin this new season of talks with Dr. Heather Flanagan-Steet. And so it's a pleasure for me to get to introduce her to you. Dr. Flanagan-Steet is an accomplished researcher, author, and instructor. She received her doctoral degree in molecular, cellular, and developmental biology from the University of Colorado at Boulder. Dr. Flanagan-Steet's research efforts have centered on defining the molecular and cellular mechanisms underlying pathogenesis of rare genetic diseases through the generation of zebrafish models. Dr. Flanagan-Steet joined the center this summer, coming from this complex carbohydrate research center at the University of Georgia, where she worked as an associate research professor. Dr. Steet serves currently as the director of the Functional Studies Lab at the GGC. If you haven't been visited and toured the new aquaculture facility and seen the little zebrafish, I'm sure that Dr. Steet will be happy to show you, and we are offering tours after this evening's talk. So again, I welcome all of you, and certainly welcome Dr. Flanagan-Steet to give our talk this evening.
If I can. Well, thank you very much. I'm certainly very excited to be here to talk to you tonight, especially during this Gene Week. And this is, in fact, my first Gene Week, so it's especially exciting. And I'm thrilled to talk to you about a topic that I'm certainly very interested in. And I think by your being here shows that you're interested in. And I really believe, and have heard it described this way many times now, that we're living in the middle of what really is a CRISPR craze. And so I think one thing we can all agree on is that throughout our existence, man has had a major impact on the nature of life on this planet. And that's true whether we think about industrialization, how we use our natural resources, certainly how our medical advances have both increased our lifespan, but also our population. But we're actually at a point in history that I think for the first time, we may be on the verge of having a tool that may allow us to actually shape life on the planet, not from the outside, but from the inside. And what I mean by that is with the advent of programmable nucleases, which CRISPR is a member of, we may eventually have the ability to change the genetic composition of every organism on this planet. And that's extraordinarily profound and really makes you take a pause and think about what that might mean. Having said that, I need to assure you that we are nowhere near being there yet. But the rate at which this technology is evolving and changing suggests that for some of you, particularly the youngest people in the audience, you actually may see this happen in your lifetime. And I think because we're not there yet, now is really the time for us to start thinking about what this might mean and asking ourselves questions like, is there any level of genetic manipulation that isn't okay? Is there anything that's going too far? Or are we okay with all of it? And I can say with some certainty that we are not all going to answer that question the same way. Certainly our own life experiences are going to come to bear on how we feel about those issues. Families that have known the difficulty of living with disease and devastating disorders may be very willing to eradicate those disorders from their future lineages. Others that don't know that difficult journey may answer that question very differently. But what's most important is that we start having this conversation now, and we all educate ourselves continually on these tools and what they mean for our future. So just to give you a sense of the rate at which this technology is evolving and changing, In 2002 and 2003, if you look in the literature, there may be 1 or 2 papers that mention the word CRISPR. By 2005, that number doubled. In 2018, you may be astounded, I am, to know that there have so far been more than 4,000 papers referencing CRISPR either in the title or the abstract. And it's predicted that there may be as much as 1,000 more before the year ends. That's remarkable, considering that the landmark paper that described CRISPR-Cas technology as an editing tool came out 6 years ago. It's absolutely astounding. So it's truly as if CRISPR is everywhere. And in fact, it invaded every aspect of our social dialogue. It is not just in the scientific journals like Science, Nature, and Cell, but it has graced the cover of Time, Newsweek. Recently, The Economist had an enormous spread on what CRISPR means for our future. So with all this information coming in, how much of it is fact? How much of it is hype? How much of it might in fact even be fiction? What I'd like to do tonight is give you some of the information you need to start discerning for yourself what's fact, what's fiction, and really think about what your answers to some of those questions might be. So what we'll do first is talk a little bit about the journey to bringing us to genome editing today, some of the major scientific advances that had happened and brought us to this place. We'll talk a bit about the tools, the programmable nucleases themselves, which of course CRISPR is the star member of. We'll talk about the state of the technologies, which are changing rapidly every single day, and a little bit about what the future might hold and some of the things that might be our responsibility to consider. So like any major medical advance or technological advance, the road to genome editing has really been paved by a number of achievements. And there's no way I can stand in the Greenwood Genetics Center and have a discussion about heredity and genes without mentioning these 2 people. Of course, Charles Darwin and Gregor Mendel are the first to really formally consider what biological variation is, what it means for evolution, and how traits are in fact inherited. But it's actually not until about the 1950s that the dream of something like genome editing even becomes plausible. And that first comes with a landmark experiment by Hershey and Chase, and they show that it's DNA, not protein, that provides the blueprint for life. And that's immediately followed, I'm sure as all of you know, by Watson and Crick using information from Rosalind Franklin and Linus Pauling to describe the exquisite helical structure of DNA. They showed how 4 simple bases, adenine, thymine, cytosine, and guanine, are organized, pointing toward the center of the structure, protected in its back by sugar phosphates that run in the opposite direction. We know that several years later, Francis Crick will also crack the code. He will show us that DNA is read in multiples of 3, and in fact, combinations of specific 3-pair nucleotides will code for specific amino acids. And while this information is fundamental and necessary of what's to come, it's not again until about the 1970s that there's 2 major changes that really get us on the road to something like genome editing. And the first of these comes from Fred Sanger. Many of you, especially the DNA aficionados, know that he developed a method called Sanger sequencing. And this is essential because it allows us for the first time to read the base pairs in a very specific order in a region of DNA. And this is essential for what's to come because without knowing the specific sequence of DNA, we can't with intention engineer it or change it. So we all know that today sequencing is much different. It's now automated and uses much more sophisticated methods. But this really sets the stage. The second thing that happens in the 1970s, in my mind, is actually the first demonstration of real genome editing. And that comes from 2 people, Christiane Nusslein-Wilhart and Eric Wieshaus, demonstrate that we can, in fact, alter the genetic composition in a living organism. They do this in a slightly different way than we do today. They use a random method, but they show that you can feed an organism a chemical mutagen, in this case called EMS. And when these male flies eat this chemical mutagen, it creates sequence variations in their sperm. And when these flies are crossed to normal female flies, they develop a whole brood of progeny carrying the sequence variation. And what Newsline, Bolhart, and Beachhouse show is that through these mutations that can often result in structural differences, we can use those differences to track back and identify all the genes necessary for a given process. They'll go on to do the same sort of thing. We call it a genetic screen in higher organisms, vertebrates, including the zebrafish. And in doing so, they'll isolate hundreds of genes necessary for all of these developmental processes. And in addition to just providing fundamental information to us, why this is key to genome editing is it actually transforms the way scientists think about how we should study gene function. And it actually inspires many others to consider what are other methods we could use to modulate the genome. And at about the same time, there's another technological advance. And in the scientific community, we refer to this as recombinant technology. And what this is a series of methods that allow us to isolate very small pieces of DNA from a cell's genome in a tube where we can change them and we can study them. And people start to wonder, well, if we can isolate these pieces of DNA, what happens when we try to put them back into the cell? And they develop a number of methods that allow us to do that. And one of the things they realized very quickly is when they reintroduce a piece of DNA in the cell, in a number of cases, it will actually stably reintegrate or jump into the cell's chromosome. It does this in a random way, so it can go anywhere in the chromosome. But when it does this, it's stably inherited as these cells divide and go through future generations. And as many of you know, this will provide the basis of what will become an enormous field of gene therapy. And there are many forms of gene therapy that are use all kinds of different methods today. But in its simplest form, what gene therapy was intended to do was take a cell that was no longer functioning because it had a non-functional gene and simply say, what happened when we add back a functioning gene? Can we in fact rescue that cell's process? And in fact, we can. And if that gene that we add back stably integrates, it will be passed on to future generations of cells. I think as many of you know, especially the clinicians and scientists in the group, gene therapy has not turned out to be quite as simple as originally intended, but certainly has led to several profound discoveries. Some of the difficulties come because several genes are simply too large to just add back to a cell, or some tissues are quite difficult to target. Having said that, we now use a variety of viral vectors to introduce those genes back into cells and tissues. That sort of technology is also very useful in research. So one of the ways in which we study a gene's function is to simply add it, either in its normal form or an altered form, back into an animal. And we can also use viral technology to do that. We can do that by injecting that piece of DNA, typically a gene and its regulatory element, back into a fertilized oocyte. If that oocyte comes from something like a fish or a frog that's going to develop outside the mother, that injected oocyte can now just grow into an adult organism carrying that extra gene. If that oocyte is from a mammal, we can actually put that engineered oocyte back into a recipient mother until that pup is born, which can then stably also carry that gene, allowing us to really study its function in the context of a developing organism. So while there are many cases where modified versions of this have been successful, even approved as therapies, gene therapy doesn't always work for all situations. As I said, sometimes the gene is just too large to add back into a cell or tissue. And in other cases, the genetic mutations actually result in something other than a non-functional protein. In that case, we have to consider additional ways to either repair or replace the existing gene. So because of this, by the early 80s, the race for more effective gene therapies was on. And as part of that, people started wondering, how could we modify genes right in their place in the genome? And two people, particularly Mario Capicci and Oliver Smithies, asked the question, if we can randomly insert DNA into the genome, why can't we intentionally have it go where we want? Why can't we intentionally target the integration to a specific gene? And this really relied on the principle that DNA is extraordinarily dynamic. It not only replicates or copies itself, it also very efficiently repairs damage to itself. The other thing DNA does is it recombines with itself. And what that means is when sequences that are similar to one another come close to each other, they may actually swap material with each other. So you see this chromosome that was originally just the green chromosome is now carrying part of the red chromosome, and the opposite is true for the other as well. Using this principle as a guide, Capicci and Smithy show that cells are provided with DNA here that's bearing the same sequence as the target that they want to hit. The DNA can actually integrate or jump into the target. And this is the basis for an enormous technology called homologous recombination that actually revolutionizes the way we ask questions about individual genes. It allows us to ask questions about how they function in cellular developmental processes, as well as what their role in different diseases might be. This also builds the foundation of what's coming with the programmable nucleases and CRISPR. It says that we can, in fact, engineer the gene. We can target specific sequences. And this is partly fueled by the fact that DNA sequencing is now automated. And with that comes several genome sequencing projects. So many of you know that once sequencing became automated, we also had major efforts to sequence our entire genome, as well as the genomes of many of the organisms we use as models. And as I said before, this is actually an essential component of engineering genes, because in order to manipulate or change a gene directly, we do have to know its precise. So this method is widely employed by scientists. It was used for nearly 3 decades until recently. And it's the way that we could actually specifically inactivate a gene. We could remove small portions or introduce sequence variations, perhaps those that were derived from a patient, and ask how that gene functioned. But unfortunately, the feasibility of this method, homologous recombination, to either repair or replace genes is hampered by one problem. And that's that although recombination occurs and genes, we can have DNA exchange information with itself, it happens at a very low frequency. So it's actually an incredibly rare event, not one that we can easily control. In fact, what I haven't told you is that our ability to engineer genes in that way in animal models relied on additional advance. And that was the isolation of embryonic stem cells. So a gentleman by the name of Martin Evans isolated a population of cells that we call ES cells, or embryonic stem cells. And they're called that because they're at the same developmental stage as cells in an early embryo. And what he showed was he could stably culture these cells and keep them in this stage. And if he targeted the cells with a piece of DNA that he wanted to engineer into a gene, what it allowed him to do was screen hundreds, even thousands of cells to find the one that has the right genetic change that he was interested in. He could then take that cell and expand it and actually reintroduce it into an embryo at a very early stage. And since the cells of that embryo and the cells in the dish were at very similar stages, they would mix together, and that change that he had engineered in the dish could then be given to future mouse pups. So I tell you this not because I think it's important at all that you understand how mouse technology works or how we create models in the lab, but to illustrate the point that until the last few years, engineering the genome in a specific way was incredibly difficult. It was laborious, it was cumbersome, and it was very, very expensive. And so there really were not that many labs that were capable of doing it. In fact, making a single genetic knockout could take years and thousands of dollars. So because of this, it was really not efficient enough to use for editing in a human being, and certainly could not be used as a therapeutic. So by the early 90s, people start asking, how can we simply increase the frequency of recombination so that we may be able to use this as a therapeutic? And people start looking at bacterial enzymes that they know both recognize DNA and cut it in a very specific way. And in that process, they realized that there are a number of proteins from bacteria that they could take pieces from to really put back together and engineer an enzyme that would do exactly what they want. And this is the beginning of what we now know as the programmable nucleases, and eventually will give rise to CRISPR. So the programmable nucleases are a family of enzymes. Nucleases are something that specifically cut DNA, and there are really two major classes. They're the protein-based nucleases and the RNA-guided nucleases. Protein-based nucleases are the zinc finger nucleases, or the ZFNs, and talins. We'll talk just a little bit about talins. And then there are the RNA-guided nucleases, which CRISPR-Cas9 is the most famous member of. Ultimately, while these two classes of enzymes work a little bit differently, the end point of them is all the same. They can be used to disrupt genes. They can be used to insert new genes. They can be used to correct genes in their position in the genome. They can be used to create point mutations or fix point mutations. And in some cases, especially CRISPR, they can be used to induce larger chromosomal rearrangement. So while they have slightly different mechanisms of function, they have several features that they have in common. The first is they're a nuclease, which means they cleave DNA, they cleave chromosomal DNA, and they do it in a very specific way. In most cases, they create what we call a double-stranded break, meaning they cut the chromosome completely across on both helical strands. When they make this double-stranded break, what it does is it actually triggers the cell to use its endogenous repair mechanisms to fix that break. And we'll talk a little bit in a minute about how that happens. The most important part of a programmable nuclease is that it's just that. It's programmable. So they need to be able to be engineered so that they will recognize the sequences we want them to recognize. So all of these nucleases are modular, and we can take them apart and put them back together so that they recognize the sequence we want to target. So to sort of simplify that, the programmable nuclease needs to act as a sort of GPS system. It needs to find the region of DNA we want to target, find it specifically. It needs to bind it, and then it needs to cut it. It typically cuts it in a double-stranded way, meaning it cuts both strands of the helix. And then honestly, the nuclease just needs to get out of the way and let the cell do its job and fix that break in a certain way. There are a number of ways the cell will go about fixing those breaks. The first of these is something called non-homologous end joining. So it turns out that a double-stranded break is really kind of catastrophic for the cell, and it needs to fix it quickly or the cell will die. So it really isn't concerned with how it puts things back together, but simply that it just glues that chromosome back together. Unfortunately, when there's a double-stranded break, it's not usually clean, so the ends aren't even, they're kind of jagged. And that's a little bit more difficult for the cell to just glue back together. What will often happen is it will clean up those ends and even them out. And one thing it can do is it can actually chew back or remove these overhangs that I've shown you in red. And then it's nice and clean, and the cell can glue that piece of DNA back together. The problem with that is when you chew those ends back, you're actually deleting sequence from that gene or that region of the chromosome, which can disrupt the gene's function. The other thing the cell might do is actually fill that region in to even out those ends. That's great. It now is clean and it can put it back together. But again, now that's created an insertion of some nucleus. And we know that since the DNA is read in multiples of 3, those deletions and insertions can change the way that gene is read and often render it non-functional. So this is actually a tremendous technology in research because it's a very rapid way for us to map out a gene's function and ask questions about it. it's not a great way to go about fixing a gene or repairing a disease mutation. So the other thing that can happen is we talked a little bit about homologous recombination and how DNA sequences can integrate in the chromosome. And it turns out if there's a double-stranded break, the frequency of that recombination significantly goes up. So what we can do is make that double-stranded break, but provide the cell with a wild-type or normal donor that it can use for repair. And if that break is made in a region that happens to contain a mutation, and the cell uses that donor to repair it, and it does it correctly, then you've actually restored the normal sequence to that gene through this method. So this is a plausible method to go about repairing disease genes. So I want to tell you a minute about one kind of programmable nuclease, and then we'll talk specifically about CRISPR. The TALENs were really the first type of programmable nuclease that was effective. These are protein-based nucleases, and this stands for transcription activator late effector nuclease. It matters not what its name means. What you need to understand about it is it's protein-based, and what that means is it recognizes the region of DNA using a module of protein. CRISPR, you'll see in a minute, uses a module of RNA. So this shows you one of those protein modules. You'll notice that it's arranged in these colorful ovals that represent different domains of that protein. And what's important about this is each of these little blocks recognizes a single nucleotide. And they really function as little blocks. We can actually, in the lab, take those pieces apart and re-put them back together in a different order so that we can engineer this to read any sequence we want. If we then hook up to this DNA recognition or binding domain to an enzyme, in this case called FOC1, which actually cuts DNA, we can actually direct where this nuclease is going to go and how it's going to cut the DNA. What's unique about the TALEN is they actually don't function with just this component, but they use two protein components. One we refer to as the left arm and one as the right arm. And that's because this enzyme that cuts the DNA has to function as groups of two. The advantage that this provides is that this molecule is recognizing a very large sequence of DNA, making it fairly highly specific. So statistically, the more nucleotides you recognize, the less likely you will find that exact sequence in more than one place. So this type of nuclease currently is still the most specific nuclease. It has a very low off-target rate, and what that means is it doesn't have a high tendency to cut sequences of DNA outside the one that you want to target. That makes them have great advantages. The problem with this type of programmable nuclease is just that it's large. So it's bulky, it's harder to get into cells than something like CRISPR is, and the fact that it has these large protein components means it's more difficult in the lab to put together than something like CRISPR is. It also means it's quite a bit more expensive, but it works and it's still used in research today. So we really had, this is a quote from one of the original creators of programmable nucleases, and we really had just gotten TALENs working, people were using them, things were going great, and then the new kid on the block comes, CRISPR. TALENs get pushed aside, and people really are most interested in using CRISPR. And you'll see in a minute why that actually is. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A little bit of a mouthful, but it'll be clear in a minute why that's its name. But before we get to that, I want to really give you a sense of why CRISPR has really kind of pushed these other nucleases aside. And that really has to do with feasibility. So I showed you with this programmable nuclease, the TALEN, that it recognizes DNA through a somewhat large protein-based component and then cuts it through this nuclease FOC1. And you'll remember that there's actually two of these pieces that come together, making it a bit large, a little bit hard to engineer, and more expensive. CRISPR is this cute and delicate. It recognizes DNA through a very small piece of RNA. RNA is very easy to synthesize quickly and cheaply in the lab. And then it has a protein component called Cas9 that will cut DNA. So it's really in the fact that CRISPR is easy to generate, inexpensive, and it's easy to get into cells, meaning it's much more feasible to use. And this is why it's really become very popular versus TALEN. In the end, CRISPR really functions very similarly to the TALEN. It recognizes a particular region of DNA, it binds it, it cuts it, it also cuts it in a double-stranded way, and then it gets out of the way for the cell to do basic repair mechanisms. It also uses non-homologous end joining, which is again an error-prone mechanism of repair and can lead to these insertions or deletions and often lead to a gene disruption. We can also do the same thing we do with TALEN, where if we provide a template for repair, and that can actually be the other chromosome, or it can be a template that we give the cell, that can facilitate what we call homology-directed repair and be used to put a wild-type sequence back in that place. Or if for research, we actually want to put a mutant in that place and study that function. So where does CRISPR come from? It didn't just come out of nowhere. It turns out that like all organisms, there's a constant battle in bacteria against would-be pathogens. Those pathogens are often viruses. And it turns out that CRISPR is actually a mechanism that bacteria use to fight off pathogens. So if you just look at this picture in the upper corner here, this is an image taken from an electron microscope. It's actually a virus. It looks a bit like the lunar landing module. I think it's terrifying looking. It's actually attached to a bacterial cell in preparation to infect that cell with its DNA. And this is how many bacterial viruses work. And from the virus's standpoint, the bacterial cell is really just a good host. It can inject its DNA, it can hijack the cell's normal mechanisms, which will allow it to replicate itself. From the bacteria standpoint, this isn't a great deal. It's hijacking its cellular processes and components. And once that virus replicates itself, it has to get out. So to get out, it often ruptures the bacterial cell, killing it. In light of that, bacteria have evolved a number of ways to ward off these viruses. Most of these ways depend on cutting the DNA that this virus is injecting up as it enters the cell. And if it cuts the virus's DNA up, it renders it non-functional, and it can't infect the bacteria. So as I said, CRISPR is actually a bacterial immune system. So it turns out that the CRISPR is actually present in the bacterial's chromosomes. This is one component of the CRISPR. So it contains a series of DNA repeats. These repeats are shown here as the black diamond. That's the short palindromic repeat parts. And these repeats are separated by what we call spacers. These spacers are not identical in sequence like the repeats are, but in fact are identical to pieces of DNA found in viruses. CRISPR, the locus, has another part to it. So this is the CRISPR part. And then there are a series of genes called the Cas genes, or CRISPR-associated genes, that are present to the left of the locus. So it turns out that CRISPR was first identified about 25 years ago by a group that was looking for another gene in the bacterial genome. And no one really understood for a very long time what CRISPR was, if it had any purpose, until computational analysis showed that these little spacer regions were indeed very similar to what we find sequence-wise in bacterial viruses. It also showed that this whole locus is actually present in the majority of bacteria we look at. So that suggests it might have some importance. It wasn't until a group of microbiologists at a food company, Danisco, that makes yogurt, were working with milk fermenting bacteria. And you can imagine if you're working with a fermenting bacteria to make yogurt and it gets a virus and your fermentation stops, you can lose millions of dollars in yogurt production. So these people were looking for a way to isolate a strain of bacteria that was resistant to viral infection so that their yogurt production would go better. And they really focused on this CRISPR locus and realized that it was indeed a bacterial immune system. So once that came out, two people, Jennifer Doudna and Emmanuel Charpentier, came together and were really interested in understanding how this immune system worked. And they showed that it used an RNA component to guide a nuclease, something that cuts DNA, to a very specific sequence, and then allow that nuclease, in this case Cas9, to cleave the DNA. So when viruses infect their DNA into bacteria, shown here, the bacteria first responds by sequestering that DNA. So it isolates it by binding a number of Cas proteins. Then, if this is a naive infection, such that the bacteria has not seen it before, it will take a piece of this DNA and insert it into the CRISPR locus. And it's been referred to as a sort of immunization card or a memory, so that in the future, the bacteria can actually fight that virus off. What then happens is this CRISPR array is transcribed, or it's converted to an RNA, a very long RNA, that contains the spacer region interspaced by something that we call a three-dimensional hairpin. And this part is expressed from these little diamonds. This long RNA is then cut up into smaller RNA components, and these are sort of scouts. They roam around the bacteria looking for those infecting viral DNA. And using this region that comes from the spacer that you'll remember was put back into the bacterial chromosome from the virus itself, it can recognize that invading pathogen and bind it very specifically. When it finds that DNA and binds it, this hairpin allows the Cas9 nuclease to join that complex and bind to it, and the Cas9 is a nuclease. It cuts DNA. And this allows it to cut the viral DNA up. Once it's cut up, it's no longer functional and can't infect the bacteria. What Doudna and Charpentier realized very quickly is if the Cas9 protease or nuclease could in fact cut DNA other than viral DNA, this would be an extraordinarily powerful tool. And so they quickly show that in addition to its ability to cut infecting viral DNA, if they engineered the RNA component to recognize a DNA that they put into the bacteria, they asked whether it would cut it up. And in fact, it did, just as efficiently as it cut up the virus. And so with that, the CRISPR-Cas9 engineering tool is born. And they make some modifications to this system. It still contains the Cas9 component, which is the nuclease that cut the DNA. They now call this a small guide RNA. So a portion of this RNA has the region that we synthesize to recognize some region of DNA we want to target. And then this region is a modified version of that hairpin that will bind and bring the Cas9 to that region and cut it. And then in cells, it's just the same as other programmable nucleases. This double-stranded break induces cellular repair. That's either this non-homologous end joining, where we have to fix the chromosome and put it back together, and that can lead to insertion of information. Or we can do this donor-mediated repair, where we give a sequence back. That can be the wild-type sequence again to fix a mutation that's there, or we can actually introduce a mutation that we want to study. So the advantage to CRISPR over these other nucleases is it's small. It's very easy to get into cells. The RNA component that recognizes the DNA portion is very easy to synthesize in the lab, and it's very inexpensive. They also have a lower cellular toxicity than some of these larger programmable nucleases. But it's not perfect. In fact, it does have a higher off-target frequency. So it will hit in sequences outside the one that we're interested in. And that's partly because this region is so small. Statistically, there are chances that you will find that same sequence somewhere else in the chromosome. Now, the technology is evolving rapidly, and people are looking at ways, obviously, to improve this. And one of the ways is to use multiple guide RNAs to really increase the number of DNA base pairs that we're trying to target, decreasing that frequency of off-target. This is not something that I'm going to go through or you need to understand, but the sort of bottom line of one of the other limitations is we can't actually, with CRISPR, target just any old sequence that we engineer. There are some constraints as to what the Cas9 can cut and what the guide RNA can recognize. So if it happens that the mutation we want to repair falls in one of those untargetable regions, CRISPR right now is not going to be a great method to do that. TALENs would work in that case. They are more difficult to work with. But again, the technology is changing, and every single day there's a paper coming out that now targets have been expanded for CRISPR because people have modified the enzyme. So it will get to where likely we can target just about every sequence in the genome. So with that, I just want to talk for a quick minute about some of the changes in the state of the technology. This is just a slide to illustrate that there are a number of different ways that the technology is being changed to do things other than just cut DNA in a specific way. And two of them that I want to tell you about very quickly are ways in which CRISPR is being used to turn genes on and off, and ways that it's being used to image chromatin. So some very astute people recognize that if we kill the Cas9, meaning we just inactivate its ability to cut DNA, it's still a great way to target something to a specific sequence. If you then hook up another kind of protein to this complex, say a protein that actually is something we call the transactivator, something that will turn a gene on, or a repressor, something that will turn a gene off, you can actually use this complex to control gene expression in a fairly exquisite way. And so if a disease actually involves changes in expression of genes other than the mutated locus, this may be a way to have sort of a downstream therapy. Another very exciting thing that people have done is use this deactivated or dead Cas9, again, as a target to regions in the chromosome, and simply hook that up to proteins that glow, fluorescent proteins. And in that way, we can ask whether specific sequences are present in DNA. We can actually use it as a probe. We can also use it to ask what the state of the DNA is. Is it in a very open conformation, letting that probe get in, or is it in a tight, closed conformation? The last thing that has happened literally in the last about 6 months is CRISPR has been changed to be a much more exquisite editing mechanism. So what we've talked about a lot is how if you direct this Cas9 protein through the guide RNA to a specific sequence, it makes a very heavy-handed kind of cut across the DNA. And while there are repair mechanisms to fix that, and it can be used to repair mutations, it doesn't always go perfectly. The repair isn't always perfect. So what people have done now is, again, use that inactivated Cas9 that no longer cuts the DNA, but give it other enzymatic activities by simply combining it with other kinds of proteins. And while I won't go through the chemistry of how this works, there are enzymes that can see certain base pairs, like adenine, and slightly change their chemical composition in their place and make them look like a different base pair. In this case, it can trick the system into thinking guanine. So if the original mutation was actually, or wild type sequence was a guanine, and the mutation made it an adenine, there are ways that you can actually reverse that. And this is a much more delicate tool than making a heavy-handed cut. This actually is a very recent addition to the CRISPR toolbox. It's happened in about the last 6 to 8 months. But now people are looking at all kinds of enzymatic activity that they can use to edit single bases within the DNA. There's also been findings that you can edit RNA, the sort of downstream component that comes after DNA. And in this case, instead of using Cas9, they're using another Cas protein from a different bacterial system called Cas13, which specifically interacts with RNA. So what are the limitations and concerns? Certainly right now, its use in research, there's really no limitations. We can tolerate in research off-target, some inconsistencies and imperfections. But in humans, we certainly can't do that. So right now, the major concern is safety. And until the efficiency of making a desired change is perfect, it really is not great as a therapeutic tool. But we will get there. The second is, because of safety, the vast majority of your CRISPR-based medical advances right now are likely to come from its uses in basic and translational research. But I assure you that that is certainly going to change. The other concern right now is, of course, delivery. So like many other therapies, we have to be able to get the CRISPR into cells and tissue, and this must be safe. And of course, right now, the best vehicle for delivery is viral-based vectors. These vectors have become very good and are fairly reliable and safe. And the other is some tissues will still remain a challenge, and until gene therapy overcomes those issues, using CRISPR to edit in more difficult areas of the human body may not be feasible for some time. So I really have been thinking about CRISPR lately as kind of like the bullet train, where things are changing at such a rapid pace, it's actually difficult to keep up with what the current state of the technology is. But I'll just show you a couple things that have actually happened. People are trying to eradicate malaria. So they've actually engineered a mosquito that cannot be infected by the malaria-causing parasites. There have been cases, particularly Layla's story, which you can look up on the internet and see, where she was treated for acute lymphoblastic leukemia. In this case, it was actually a TALEN engineering that they altered T cells and allowed it to fight the leukemia, and now she is in complete remission. Scientists have shown in rats that they can treat blindness by reversing the mutation that caused retinitis pigmentosa. And again, if there was some off-target rate in these rats, that can be tolerated in the lab. That can't be tolerated in a human being yet. So we're not at the point of using the technologies, but the proof of principle is actually there. The other place we're seeing a lot of advances is in agriculture. So some of you may be excited that there's a gluten-free strain of wheat coming. There are tomatoes that have long shelf lives. And while many people have different issues with genetically modified food, one thing I can say that could be an advantage here is with recent events like the tsunami, this might actually allow us to get fresh food to people after disasters before the food rots. People are also working at making pigs that we can grow human organs in for xenotransplantation, where your body would then not reject the organ because it's actually a human organ. And one of the recent things that happens is there's evidence that we can use CRISPR to treat Duchenne muscular dystrophy. This did not reverse Duchenne muscular dystrophy, but it was really a proof of principle where people took a virus carrying a CRISPR, injected it into beagles that had muscular dystrophy, and showed that in some of their muscles, this is a normal muscle, this is a Duchenne muscle, some of their muscles actually reverted to what looks like a normal muscle. There's also the wild and crazy. Someone has created a mini pig for sale as a pet. Others are trying to use CRISPR in elephants to revive the woolly mammoth. You can all decide for yourself if you think that's an important evolutionary study. I won't comment. CRISPR is absolutely everywhere, though. I think there is no doubt that it's in every aspect of our lives, and this train is moving and it's not stopping. One place that it is not, however, and I truly assure you, it is not here. We cannot create engineered babies. There are so many reasons that we are not able to do it. The first is the technology is not nearly sophisticated enough yet to do this. The second is that the traits that lead to a human being are not due to one gene or two genes. These are very complex pathways giving rise to things like vision and IQ. So we're nowhere near being able to do something. So I would tell you when you read these articles, a good deal of it is not fact and a good deal of it. What I want to leave you with is the idea that we are really just at the tip of this iceberg. This is a tremendously powerful set of technologies, and as it becomes adapted, it should be incredibly useful for all kinds of things, including medical therapies. But what we need to remember throughout all of this is that for every iceberg, there's what we can see above the water, and then there's a very large additional iceberg below the water that we can't. So for everything we do, there may be unintended consequences that we can neither predict nor see coming. And those are the sorts of things that we may want to start thinking about in advance, how we would deal with these and what we're comfortable with. So with that, I really thank you for your attention for quite a long time and take any questions.